Hello everyone. Welcome to this another session in this national workshop on biostatistical analysis. Today with us we have Dr. K.V. Bhat. Dr. K.V. Bhat is Professor Emeritus at NBPGR, IARI, New Delhi. He has done his PhD in genetics, cytogenetics, plant breeding and plant pathology from College of Agriculture, University of Agricultural Sciences, Karnataka. He has been in active research and teaching at this institute. His area of expertise and research lies in DNA fingerprinting and bioinformatics. He has guided 5 MPhil students and 10 PhD students. Dr. Bhat is an illustrated scientist and has more than 100 research publications in various national and international journals of repute. He has been invited as a resource person in many national and international workshop and training program. So we are delighted to have a talk from Dr. K.V. Bhat. The voiceover in the video is given by Dr. Ruchi Veer. Dr. Ruchi Veer is Assistant Professor in Zakir Hussain Delhi College, Delhi University. So I hope you will enjoy this session. Thank you.
Topic of this video is Analysis of Molecular Variance by Dr. K.V. Bhat and voiceover is by Dr. Ruchi Veer. We are mostly interested in determining the extent and nature of genetic diversity among individuals representing subgroups or groups. The studies also involve defining differences between the individuals within and between groups. The information thus derived help in various genetic diversity conservation, management and utilization activities. It tells us what are the highly diverse group, how much is the extent of diversity in them, how the diversity is distributed among them. This is essentially related to temporal and spatial distribution of diversity. In population terms, the groups might be representing population, subgroups and the subpopulation, and several population might be representing a metapopulation. These studies and answers to these questions are required to plan our biodiversity management activities. Now, before proceeding further to the topic, it is essential to define certain terminologies that we use, and the first terminology that comes in our mind is what is a population. So a population is local group of conspecific organism sharing a common gene pool. Hence, members of a population have a capability to interbreed and hence share a common gene pool. In this example, which I typically encountered during plant collections, we have a river with 3 tributaries A, B and C. Obviously, the localities through which these tributaries pass are geographically isolated. Hence, the representatives of the plant species of interest to us that occur here are isolated, even though they have capability to interbreed. Hence, share the gene pool. This might be true for upstream members of the species, but downstream it is likely that they get derived continuously from upstream members in addition to the greater presence of progenies of members that got established earlier. The question to answer here is whether the member are to be considered to belong to 3 different groups or populations or subpopulations What is the extent of sharing of alleles A, B and C, groups or subgroups? Extent of flow of genes in each generation, how strong is the extent of differentiation of groups, etc. Now, the answer could be A, B or C. Before planning an activity towards diversity management, we need to be clear whether we are looking at 3 populations, 2 populations or only one population. The figure here explains defining just 3 of the possibility, though several others exist. Even though the statistical measures are listed here, that are measures of genetic diversity, that is NA, number of alleles per locus, NP, number of polymorphic loci, HO, heterozygosity observed, HE, heterozygosity expected. These statistics helps us in determining the extent of genetic variation residing in each of the putative groups or subgroups. Now, even though the statistical measures listed here help in the question of defining subpopulations and populations when taken together rather than individually, some of them are more informative than others. Combined, these measures provide substantial information on genetic diversity and differentiation of groups. However, it is important to know and consider that the basis of these measures are in the population genetic principle of Hardy-Weinberg equilibrium. Now, heterozygosity is a key measure of diversity. The Hardy-Weinberg population equilibrium provides us with a way to estimate a key measure of genetic diversity, H. Now, what is H? H is a relative frequency of heterozygotes in a random mating population, also denoted as HO for observed, the expected amount of heterozygosity at a locus. HE is derived from the frequencies of alleles at a locus. Now, a comparison of HO and HE tells us about the nature of mating taking place among the individual in a population. If HO is equals to HE, there is random mating occurring. However, if HO is greater than HE, outcrossing is the preference. And the other case, when HO is less than HE, selfing or crossing between the individuals is preferred. The scheme here explains how 3 genotypic frequencies are derived and estimated. And the formula explains relationship between the homozygotes and heterozygotes in a population under random mating. Hence, the sum of frequencies of 3 genotypes is always 1. Now, coming to the next slide about Wright's F statistics. So, a Wright F statistics or inbreeding coefficient for a single population, reduction in heterozygosity relative to random mating population with the same allele frequencies. The estimates of heterozygosity in different level of population are used to define the quantum of inbreeding in the subpopulation as FIS, FIT and FST. The concepts were defined by Sewell Wright as various F statistics. The implications of these are explained in our next slide. So when we talk of FIS, that is F inbreeding coefficient, and it is used for estimating correlation of uniting gametes relative to gametes drawn at random from within a subpopulation averaged over subpopulation, whereas FIT, or you can say F total inbreeding coefficient, it explains correlation of uniting gametes relative to gametes drawn at random from the entire population. Then the third coefficient, that is FST, or co-ancestry coefficient, it explains correlation of gametes within subpopulation relative to gametes drawn at random from the entire population. Now, these slides explain the meaning of all three F statistics commonly calculated and used to quantify the extent of population subdivision. So, when we talk of F statistics are independent of allele frequencies and are function of the mating system. If the mating is random within the population, FIS, that is the first coefficient, it is 0. However, if the mating is random within the subpopulations and allele frequencies are equal among them, then the FSTs is equals to 0. Now, the next thing that we have to focus on are the FST statistics, that are the fixation indices. Now, the measure of FST is commonly used to determine the extent of differentiation the subpopulation might have undergone, since it quantifies the average reduction in heterozygosity among two subpopulations relative to total combined population. Please note here that heterozygosity in subpopulation, that is HS, is estimated from Hardy-Weinberg estimation principles, and HT is derived from the average frequencies of the alleles P and Q at a locus. Hence, FST is the proportion of difference in heterozygosity in subpopulations in relation to total population under Hardy-Weinberg equilibrium. Note here that depending on number of designated subpopulations, the H are estimated and averaged out. Now, the next slide is about how can we interpret the FST. Now, as you can see, the theoretical range of FST is from 0 to 1, but in reality, it is usually much smaller than 1. Now, the interpretation of FST estimates are subjective. However, it may be noted that this thumb rule can be used in general, although depending on the previous estimates of genetic differentiation in the species, the interpretation of the degree can be modified. This means, though in most instances 0.25 or higher indicates presence of very great degree of differentiation of subpopulation, in some specific instances even 0.15 can be interpreted to indicate presence of very great degree of differentiation. This must be substantiated by estimates using other parameters as well. Now, the next thing that we will be discussing about is the effects of population substructure. Now, the effects of population substructures as they are listed above includes reduction in heterozygosity, genetic divergence of subpopulation from one another, which might be due to any of the evolutionary processes such as selection, reduction in gene flow, and genetic drift. Please note that these processes result in reduction or variation in allele frequency. The phenomena of random genetic drift and its consequences are explained in next slide. Now, what is genetic drift? Random fluctuations of allele frequencies due to sampling of gametes at reproduction. Alleles are fixed or lost from the population simply by chance. Without gene flow, every allele in every population will eventually become lost or fixed. However, the rate of drift is much faster in the smaller populations. Random genetic drift is entirely a random process and not due to existence of other evolutionary forces. Hence, allele frequency can fluctuate in a population over generations in a random manner. It can result in fixation of alleles at random in a population or loss of alternate alleles as shown in the graph. Its effect is influenced by population size as indicated by the number 2n, where n is the number of individuals in a population. So, in the populations with less than optimum number of individual, that is 2n is equals to 18, drastic changes in the frequencies are observed as compared to large population where 2n is equals to 100, where the changes in allele frequencies are minimal. Note that in first population with 2n is equals to 18, in 4 generations at least one population has experienced fixation of an allele, thereby leading to loss of genetic diversity. Further, it can be noted that the changes in the allele frequencies are drastic over generation. In the population, it may change from 0.5 to 0.1 in one generation, but again in the next generation the frequency can be 0.8. A hypothetical stream network you are observing on your screen. We see here a worked example of earlier mentioned hypothetical network of stream and a river with 3 putative subpopulations. The frequencies of 2 alleles at a locus scored in these 3 subpopulations as P and Q are given. Note that total of the 2 frequencies P and Q in a subpopulation is always 1. The 2 PQ estimates the proportion of heterozygotes. The frequency of P of one of the alleles varies from 0.2 to 0.6, while H varies from 0.3 to 0.48. These estimates can be used to quantify the extent of subdivisions of the 3 subpopulation as FST. The calculated FST is 0.11 in this case. As per the general thumb rule, it indicates the presence of moderate differentiation of subpopulation. Now we will be talking about the population substructure. So many species, they naturally subdivide themselves into herds, flocks, colonies, schools, etc. The reasons for population subdivision as a consequence of existence of population substructure are explained here. It can exist in many species which naturally subdivide into herds, flocks, colonies, schools, land races, varieties, etc. The presence of patchy environments as indicated by variations in fertility, moisture, altitudinal gradients, fragmentation of habitats, geographical isolation, factors influencing random mating all lead to population substructure. Population subdivision leads to reduction in heterozygosity and generates genetic differentiation through natural selection and genetic drift. So this is a classical example where we sample a mouse population that has become subdivided in a barn and look at a single locus with 2 alleles in the population, that is AA. This hypothetical example explains influence fragmentation of habitat on subdivision of population. The subject here is a population of mice with 2 alleles, capital A and small a, in a large barn with 2 walls separating 2 groups. They are connected by 2 small doors, but the space between are inhabited by predatory cats which devour the mice trying to cross over to the other subgroup. The allele frequencies P is equals to Q is equals to 0.5 initially. However, this consequences of this fragmentation of habitat is explained further. After several generations, these 2 subgroups are analyzed by sampling them by catching the mice straying into the central wing. Analysis of west and east wings separately indicated absence of alternate alleles. Thereby, west wing had only A with P is equals to 0.1 and east wing had only small a with Q is equals to 1. However, if a pooled analysis is made, it indicated the frequencies of capital A is equals to small a or P is equals to Q, that is equals to 0.5. But note that there is complete absence of heterozygotes, that is 2pq is equals to 0. The complete absence of heterozygotes and presence of very high frequencies of homozygotes indicates the presence of great subdivisions of populations. Now, population structure, the frequency of recessive allele in desert snow flowers, that is Lananthus paraoe. An An application of this concept is detailed by Dobzhansky and Epling 1942 in their study of desert plant population. They have seen that this plant produces blue flowers when in recessive condition, thereby making it easy to score large populations. The region was surveyed every 30 miles from east to west. The estimates of H are plotted for each subpopulation sampled. We can clearly observe the presence of differences in the estimates. The west had higher values, that is 0.302 to 0.717, and east had lower estimate of H, that is varying from 0.014 to 0.411. Further, the central zone has extremely low estimates of H. The averages for each region were 0.499, 0.027 and 0.0306. This estimate of region's fixation index was 0.1589 and overall fixation index of 0.329. This example provides utility of this procedure in the study of population subdivision. Now, moving forward, this slides of hierarchical structure of the desert plant adapted from Wright 1943 explains how calculations are made for each of the statistics. Note that HT here is estimated from population frequencies of alleles P and Q. Now, this means example for the desert plant where FST is equals to 0.2371 minus 0.1424 divided by 2371. Overall reduction in average heterozygosity is close to around 40% of the total heterozygosity. Subpopulations of this species are highly differentiated. This particular slide on your screen shows the various software that are available for analysis like PopGene along with their URLs and functions, and this list of several software that can be used to calculate allele frequency from molecular data including DNA sequences. The links from where these can be downloaded are also given under URLs. The objective of this presentation is to provide overview of the statistical analysis of data on molecular diversity to partition the total variation into within group and between group variations and provide a way to test their significance statistically. The most commonly used software is the Harlequin, which is detailed in further slides. So this a versatile software for AMOVA, that is Harlequin. It is the most commonly used software for partitioning of molecular variants and partitioning it into various sources is Harlequin from L. Excoffier, which is detailed in slides that are coming on your way. Now, analysis of molecular variance. As the name suggests, analysis of molecular variance, that is AMOVA, is a method for studying molecular variation within a species. Electrophoresis, one of the most widely used method for studying the structure of DNA, produces data in the form of zeros and ones, where 1 denotes the presence of a band and 0 is its absence. For example, AMOVA works on such data to create distance matrix between samples in order to measure the genetic structure of the population from which the samples are drawn. In statistical terms, AMOVA is a testing procedure based on permutational analysis and involves few assumptions about the statistical properties of the data. Though here we are addressing analysis of one type of qualitative data procedure, same procedure can be used for AMOVA of genotypic, that is AA, AB and BB, and DNA sequence ATGC data. Now, next is analogy with ANOVA. Now, AMOVA is like an hierarchical analysis of variance in that it separates and tests tiers of genetic diversity. Diversity among groups of population. Number 2, diversity among populations within group. Number 3, diversity among the individuals within a population. When studying molecular variation, it is customary to examine haplotypes, and so there is no variation within individuals. AMOVA differs from analysis of variance in that AMOVA can accommodate different evolutionary assumptions without modifying the basic structure of the analysis. Hypotheses are tested using permutational method so that no normal assumption is required. Now, moving to the next slide, that is AMOVA analysis of molecular variance. It is method of estimating population differentiation directly from molecular data, that is, for example, from RFLP, direct sequence data, or phylogenetic trees. The variance components are used to calculate phi statistics, which are analogous to Wright's F statistics. Now, moving on to the next slide that explains AMOVA in bit detail. Hierarchical fixation indices are conducive to inclusion of more levels. Before we had individual, subpopulation, or total population. We could instead have the following: individual, population, group of subpopulation, or entire population, that is equals to all subpopulations. This can be properly addressed with AMOVA. Now, AMOVA and examples of hierarchical levels. Fixation indices can be calculated for among group of populations that are indicated by red lines FCT, and among subpopulation within groups indicated by green lines, that is FSC. This is done within an analysis of variance framework using covariance components within a general linear model. So various fixation indices provided by Harlequin are therefore here they are explained. Now moving on to the next slide. So this represents an AMOVA table. AMOVA table here indicates how the partitioning of total variation is done. Here, as in case of ANOVA, the partitioning of variance is into among groups, among population, or within groups and within population in the hierarchy. The within population is the lowest level and among group is the highest level of hierarchy. Now, the variances due to each of the sources are used to derive the estimates of variances within the population, among population and among groups as SSD, WP, SSD, that is AP divided by WG, and SSD, that is AG respectively. As per expectation of mean squares column, these estimates are used to calculate equivalent of FST, here indicated as FCT, for comparing total with groups. Now moving on to the next slide, that is significance of variance components. The meaning and interpretation of each estimates provided by Harlequin is summarized here. Permutation analysis depending upon the component. For FCT, this is typically done by permuting population among group. For FSC, this is done by permuting genotypes among population within group. And for FST, this is done by permuting genotypes among population among groups. Now, the next slide, it's an input data file and it shows that this is an example of several input data files. This is for the DNA sequence data. The software upon installation unzips the stores various input data files in a separate folder within the main folder. Use of these models input files are advisable to practice use of Harlequin. The headings and the end portion of each type of file reduce the problem in formatting the input file. These can be opened using Notepad and edited data copy paste for easy use. So the next slide that is on your screen, it shows the gist of capabilities of Harlequin, which makes it the most appropriate for such analysis is summarized. Further, provision to integrate it with dedicated R scripts helps in generation of publication quality, graphics and plots. So thank you very much.